DNA repair mechanism. Since most mutations are deleterious, DNA repair systems are vital to the survival of all organisms. If DNA repair systems did not exist, spontaneous and environmentally induced mutations would be so prevalent that few species, if any, would survive. In humans, an individual who is defective in only a single DNA repair system may manifest various disease symptoms, including higher risk of skin cancer. This increased risk is due to the inability to repair UV-induced mutations. The living cells contain several DNA repair systems that can fix different types of DNA alterations. Each repair system is composed of one or more proteins that display specific roles in repair mechanisms. In most cases, DNA repair is a multi-step process. First, one or more proteins in the DNA repair system detect an irregularity in DNA structure. Next, the abnormality is removed by the action of DNA repair enzymes. Finally, normal DNA is synthesized via DNA replication enzymes. Then, types of DNA repair mechanisms. Direct reversal repair. Direct reversal repair is when an enzyme recognizes an incorrect alteration in DNA structure and directly converts it back to a correct structure. An example is repair by simple conversion of damage is photoreactivation. Photoreactivation directly reverses the formation of pyrimidine dimers that result from ultraviolet radiation. In photoreactivation, the enzyme DNA photolase captures energy from light and uses it to break the covalent bonds linking adjacent pyrimidines. Another example is direct reversal is the removal of methyl group from the methylated base O6-methyl guanine. In this case, the methyl transferase removes the methyl groups from the guanine residues by transferring it to one of its own cysteine residues. This is costly to the cell because the methyl transferase is not catalytic. Having one accepted a methyl group, it cannot be used again. Then mismatch repair. The structure of DNA double helix obeys an ATGC rule base pairing. During the normal course of DNA replication, however, an incorrect nucleotide may be added to the growing strand by mistake. This produces a mismatch between the nucleotide in the parent and the newly made strand. Various DNA repair mechanisms can recognize and remove this mismatch. DNA polymerase has 3' or 5' proofreading ability that can detect mismatches and remove them. However, if this proofreading ability fails, cells contain additional DNA repair systems that can detect base mismatches and fix them. An interesting DNA repair system that exists in all species is the mismatch repair system. The molecular mechanism of mismatch repair has been studied extensively in E. coli. 3 points designated mut-S, mut-L, and mut-X detected the mismatch and directed the removal of the mismatched base from the newly made strand. These proteins are named MUT because their absence leads to a much higher mutation rate that occurs in normal strains of E. coli. The role of MUTS is to locate mismatches. Once a mismatch is detected, MUTS forms a complex with MUTL. MUTL acts as a linker that binds to MUTH by looping mechanism. This stimulates MUTH, which is bound to a hemimethylated site, to make a cut in the newly made non-methylated DNA strand. After the strand is cut, MUTU, which functions as helicase, separates the strands, and an exonuclease then digests the non-methylated DNA strand in the direction of the mismatch and proceeds beyond the mismatch site. This leaves a gap in the daughter strand that is repaired by DNA polymerase and DNA ligase. The net result is the mismatch has been corrected by removing the incorrect region in the daughter strand and then resynthesizing the correct sequence using the parental DNA as a template. In excision repair, there are two types of excision repair mechanisms. Base excision repair. Base excision repair involves the function of a category of enzymes known as DNA and glycosylases. This type of enzymes can recognize an abnormal base and cleaves the bond between it and the sugar in the DNA backbone, creating an apyrinic or apyrimidinic site. So, living organisms produce multiple types of DNA and glycosylases, each recognizing particular type of abnormal base structures. Depending on the DNA and glycosylase, this repair system can eliminate abnormal bases such as uracil, 3-methyladenine, 7-methylguanine, and pyrimidine dimers. Base excision repair is particularly important for the repair of oxidative DNA damage. Here, the DNA contains uracil in its sequence. This could be happen when spontaneously or by the action of chemical mutagen. There is uracil within the DNA and cleaves the bonds beyond the sugar and base. This releases the uracil base and leaves behind an apyrimidinic site. This abnormality is recognized by a second enzyme called AP endonuclease, which makes a cut on the 5' end. Following the cut by AP endonuclease, one of three things can happen. In some species such as E. coli, DNA polymerase 1, which has a 5' or 3' exonuclease activity, removes a DNA segment containing the abnormal region and at the same time replaces it with normal nucleotides. This process is called a nick translation, although DNA replication, not mRNA translation, actually occurs. Alternatively, in eukaryotic species such as humans, the DNA is repaired in two possible ways. DNA polymerase beta has the enzymatic ability to remove a site which is missing base and then insert a nucleotide with the correct base in its place. Another possibility is that DNA polymerase delta or epsilon can synthesize a short segment of DNA which generates a flap. The flap is then removed by flap endonuclease. Then nucleotide excision repair. An important general process of DNA repair is the nucleotide excision repair or NER system. This type of system can repair many different types of DNA damage including tiny dimers, chemically modified bases, missing bases and certain types of crosslinks. In NER, several nucleotides in the damaged strand are removed by the DNA and the intact strand is used as a template for resynthesis of a normal complementary strand. NER is found in all eukaryotes and prokaryotes, although its molecular mechanism is better understood in prokaryotic species. In E. coli, the NER system requires four key proteins designated UVRA, UVRB, UVRC, and UVRD, plus the help of DNA polymerase and DNA linkase. UVR, ABC, and D recognize and remove a short segment of damaged DNA strand. They are named UVR because they are involved in ultraviolet light repair of pyrimidine dimers, although the UVRA-B proteins are also important in repairing chemically damaged DNA. A protein complex consisting of two UVRA molecules and one UVRB molecule tracks along the DNA in search of damaged DNA. Such DNA has a distorted double helix which is sensed by the UVRA-UVRB complex. When a damaged segment is identified, the two UVR proteins are released, UVRA proteins are released, and UVRC binds to a site. The UVRC protein makes cuts in the damaged strand on both sides of the damaged site. Typically, the damaged strand is cut 8 nucleotides from 5' end of the damage and 5 to 4 nucleotides away from the 3' end. After this process, UVRD, which is a helicase, recognizes the region and separates the two strands of DNA. This releases a short DNA segment that contains the damaged region, and UVRB and UVRC are also released. Following the excision of damaged DNA, DNA polymerase fills in the gap using the undamaged strand as a template. Finally, DNA ligase makes the final covalent connection between the newly made DNA and the original DNA strand. Then recombination. Of the many types of DNA damage that can occur within the living cell, the breakage of chromosome called DNA double-strand break is perhaps the most dangerous. DSP can be caused by ionizing radiation, X-rays or gamma rays, chemical mutagens, and certain drugs used for chemotherapy. In addition, reactive oxygen species that are in byproducts of aerobic metabolism can cause double-strand breaks. The two main types of recombination repair are homologous recombination repair or HRR. Homologous recombination repair, also called homology-directed repair, occurs when homologous DNA strands, usually from a sister chromatid, are used to repair DSP in other sister chromatid. First, the DSP is processed by the short digestion of DNA strands at the break site. Then this processing event is followed by the exchange of DNA strands between the broken and unbroken sister chromatids. The unbroken strands are then used as templates to synthesize DNA in the region where the break is occurred. Finally, the crisscrossed strands are resolved, which means they are broken and they rejoined in a way that produces separate chromatid. The non-homologous end joining or NHEJ. During non-homologous end joining, the two broken ends of DNA are simply pieced back together. This mechanism requires the participation of several proteins that play key roles in the process. First, the DSP is recognized by end-binding proteins. These proteins then recognize additional proteins that form a cross bridge that prevents the two ends from drifting apart. Next, additional proteins are recruited to the region that may process the ends of broken chromosome by digesting particular DNA strands. This process may result in the digestion of a small amount of genetic material from this region. Finally, any gap is filled by DNA polymerase and the DNA ends are linked together or ligated together. Then SOS response. Despite having multiple repair systems, sometimes the damage to an organism's DNA is so great that the normal repair mechanisms just described cannot repair all the damage. As a result, DNA synthesis stops completely. In such situations, a global control network called SOS response is activated. Save our soul system. So the prokaryotic SOS system is regulated by two main proteins, LEX A and REC-A. The most common cellular signals activating the SOS response are regions of single-stranded DNA, ssDNA, and arising from the stalled replication fork or double-stranded breaks, which are processed by DNA helicase to separate the two DNA strands. During normal growth, the SOS regions are negatively regulated by LEX-A repressor protein dimers. For initiation of SOS response, REC-A protein binds to ssDNA in an ATP hydrolysis-driven reaction, creating REC-A and ssDNA filaments. And REC-CA binds to single to double-stranded DNA breaks and gaps generated by cessation of DNA synthesis. REC-A binding initiates recombination repair. REC-A ssDNA filaments activate LEX-A autoprotease activity, which ultimately leads to cleavage of LEX-A dimer and subsequent LEX-A degradation. The loss of LEX-A repressor induces transcription of the SOS gene and allows for further signal induction, inhibition of cell division, and an increase in the levels of proteins responsible for damage processing. The first SOS repair mechanism to be induced is nucleotide excision repair, whose aim is to fix DNA damage. So the first SOS repair mechanism to be introduced is nucleotide excision repair, whose aim is to fix DNA damage without commitment to full-fledged SOS response. SOS repair system operates only under potentially lethal conditions caused by extensive UV radiation. Therefore, SOS repair system works on a principle that survival with mutation is better than no survival at all. This repair is also called error-prone repair. Low level of expression, and DNA damage induced signal, REC-A protein activation, REC-A repressor cleaved, then signal, then activated REC-A, LEX-A, and DNA repair and other SOS functions. DNA repaired, drop in level of signal, drop in level of activated REC-A, LEX-A repressor accumulates.